Let's start our discussion of the endocrine system with an overview. And there are three main principles I'd like to discuss. And these are principles that are going to apply in every video that follows regarding the endocrine system. And the first is, what is the main function of the endocrine system? And the function of the endocrine system is going to be to coordinate the activity of groups of cells. And that coordination might be in maintenance of homeostasis. For example, the endocrine system is going to be involved in maintaining relatively stable levels of blood glucose. But there are going to be times where the endocrine system also coordinates changes in our body, things like growth and development or regulation of stress levels and sleep-wake cycles. So the endocrine system is going to be involved in all kinds of processes in the human body that require regulation of groups of cells together as tissues. The second principle I'd like to discuss is that the endocrine system acts through hormone receptors. It's true, the endocrine system acts through hormones and hormone receptors, but I want to focus today on hormone receptors, and I hope the rationale will be clear by the end of the video. And the endocrine system consists of a collection of sensors, control centers, and effectors. So the endocrine system is going to work in feedback loops if you refer back to the feedback loop video in our Foundations of Physiology section. Okay, so let's start with the coordination of the activity in groups of cells. We have a lot of cells in our body, and the cells need to work together as tissue. For example, let's take liver cells. Liver cells will store glucose when blood glucose levels are high, or release the stored glucose into the bloodstream if blood glucose levels drop. So you can imagine if one cell is busy storing energy and the other cell is busy releasing energy, that's not going to do our neurons a lot of good, those neurons that depend on the steady blood glucose levels. So we need some system so that these cells are acting together. Now, one of the advantages we have in our bodies is all cells share a blood supply. If there are molecules that are released into the bloodstream, then they're going to be carried to cells all over the body. So this is a capillary with some red blood cells. And in green, we have some molecules floating in the plasma that are meant to represent hormones. Let's say they're proteins or steroids. And they're carried in the plasma, or they're dissolved in the plasma, or maybe carried by proteins. And then this is not to scale. Remember, these proteins are much, much smaller than the red blood cells, but just to make them visible, we'll draw them there. And then those hormones that are in the bloodstream can diffuse out of the bloodstream into the interstitial fluid to be in the neighborhood of cells. And on their own, the hormones aren't going to affect activity in the cells. The hormones are signaling molecules. They're not enzymes. They don't carry activity in and of themselves. So what we'll see is no matter what happens, if the hormones are in the interstitial space, nothing is happening inside the cells. That changes if the cells have a hormone receptor. Now, let's move along and talk about hormone receptors. Each of these cells has many, many receptors. I've depicted receptors on the cell's plasma membrane, so the hormone doesn't have to get into the cell to have an effect. It can bind to the hormone receptor on the outside of the cell. But let's look at what happens in each one of these cells in a single receptor, and then hopefully you can generalize that to what's going on in many, many receptors at once in these cells. So we're just going to look at the one cell here, and we're going to look at hormone binding to the receptor. The receptor changes shape. Now keep in mind, a hormone receptor is an enzyme. A hormone receptor is a signaling molecule that's going to change chemical reactions within a cell. For an enzyme, If an enzyme changes shape, it changes function. Okay, again, hormone binding initiates a shape change because the hormone receptor is some type of enzyme, some signaling molecule. This is going to have an effect in a cell. Shifting from off to on is going to cause some signal within a cell. So I just drew a lightning bolt and stuff happens. The particulars of what happens is going to depend on the hormone receptor. We might have different hormone receptors for the same hormone. For example, epinephrine might bind to beta-2 receptors to cause smooth muscle relaxation, or alpha-1 receptors to cause smooth muscle constriction. So the effect of epinephrine is due to the activity of the hormone receptor, not the hormone itself. I'm sure you've been in anatomy and physiology long enough now to realize that a hormone receptor that's off isn't doing absolutely nothing, but we don't need to get into those details. We're thinking of a hormone receptor as a switch. When a hormone binds, it causes a change in shape, a change in function, and initiation of a signaling cascade. I want us to think that receptor activity alone is enough to cause a change, even if a hormone's not there. So for example, in certain cancer cells, there are mutations in hormone receptors that leave them in the on state, and there's a continuous signal within those cells. For example, the epidermal growth factor receptor initiates a signaling cascade that includes cell division. So a number of cancers have a mutated receptor that's stuck in the on position. If that receptor is in the on state, even without a hormone, the cell receives signals that the hormone is present. And that's a reason I really want to focus on hormone receptor activity rather than the hormone itself when we look at the function of a hormone. Well, there are two reasons. One is I want to think that different receptors have different activities. And the second is what really matters is whether the receptor is turned on or off. But even more than that, even if the receptor is present, the cell depends on an intact signaling cascade. So insulin might bind to an insulin receptor, cause a shape change in that receptor, but some of the signals downstream might be broken, so the switch is ineffective. And we'll see an example of that in type 2 diabetes, where in type 2 diabetes, there may be a shortage of insulin receptors, so a shortage of insulin signaling, or the signals downstream of the insulin receptor might be broken. Okay, if the activity of a hormone depends on a hormone receptor, then we can call target tissue for a hormone that tissue that has receptors for the hormone or functional receptors for that hormone. So let's move on and talk about that target tissue. And I'd like us to think of the target tissue as an effector. And so we're all of a sudden talking about feedback loops. We're thinking of loops with sensors, control centers, and effectors, and the target tissue is going to be an effector. Hopefully this slide as it unfolds will look familiar. It's from our video on feedback loops. But in our body, we have certain starting points and ending points. If we're maintaining homeostasis, then the start and end point are the same. For example, if blood glucose gets out of whack, we need to initiate a negative feedback loop so that the end point of that feedback loop is steady blood sugar. On the other hand, we have some feedback loops where the ending point is different than the starting point. Like, for example, we may want to coordinate growth of bones or muscles. At any rate, in all of these feedback loops, we're going to have a sensor, control center, and an effector. And the target tissue of a hormone is going to be an effector. It's going to be the tissue that affects a change and brings us to a different ending point. So at this point, the ending point is also monitored by the sensor, so it becomes a new starting point. At any rate, why am I harping on this? Because a lot of times in a lot of textbooks, you'll see the word sensor here replaced by the word receptor. Recall that I used the term receptor to talk about a hormone receptor, which is part of the target tissue. All cells in the body have lots of different receptors for lots of different molecules. Because we use the term receptor so frequently to talk about proteins on the cell surface, I want to avoid confusion and use the term sensor for this component of the feedback loop. And in fact, what I'd like to do is make this even clearer so that we know we're talking about sensor tissue and control center tissue and effector tissue. All of these tissues consist of different cells, and all the cells have receptors. When we talk about a hormone receptor, this is in the effector tissue in a feedback loop. Okay, so let's try to summarize what I hope I've covered today. The function of the endocrine system is to coordinate the activity of groups of cells so that they work together in tissue. Now, that goal might be to help us maintain homeostasis, or the goal might be to coordinate changes within our body. The endocrine system acts through hormone receptors. Hormones are released into the bloodstream, they circulate throughout the body, and they will affect all cells that have a functional receptor for that hormone. It's the hormone receptor that mediates the activity of a particular hormone. Now, finally, each of these endocrine factors participates in feedback loops. Those feedback loops contain sensors, control centers, and effectors. Tissue with hormone receptors is called target tissue, and that tissue functions as an effector in a feedback loop. We'll talk about the different sensors and control centers as we move further in this discussion.